I wanted to quickly make an announcement that now we have the time set for PLF sessions. It'll be Wednesdays, 4:30 to 5:20 in Chaparral 5230, that second floor of this building. And then Monday, 1:30 to 2:20. The room is different though, 5227, second floor as well, but it's a different room. These slides are going to be posted in Canvas later, so you can always check the information there. And we're starting tomorrow. For those of you who might be interested in attending one of these PLF sessions, see what they are about. I didn't really explain them in too much detail, but we have some activities prepared for you in advance. Those will take about 15-20 minutes. And then it's just free time for you to ask any questions you may have about lecture, about the reading material, assignments, quizzes. Any questions from anyone on that note? No? Okay. All right, so today we'll get started with molecular genetics, with the processes of transcription and translation. And I will, as usual, try and start with let's see, there you go. Start with one of those questions that are meant to get those fluids running. What is a gene? So think for yourselves what you think a gene is, then share it with a partner, and then let's share it together as a group. Simple question. What is a gene? I'll give you a couple minutes. What is a gene? A gene, anyone?
A part of the DNA strand?
Part of DNA.
A segment that you're separating to observe?
Part of DNA / segment. To observe, that's how or why genes evolved.
It's for for a specific protein, it's going to be using that.
So it's not about us observing these segments of DNAs, what function they play, right? And what is the function of a gene? Code for protein, so code for protein. What might be a slightly different take on what a gene is, what its function is. And I'm not saying that's not true. Code for protein, since. Yeah, so let me paraphrase that. It's linked or associated with some function. Okay, phenotype is something that, again, us as external observers, we assign a phenotype, but from the point of view of the individual that has that gene, plant, animal, it's a function, right? Sure. So code for protein, let's say, carries a function. What else? What else do we associate gene with? Yeah. So we're still in the same theme, right? Expressing a function, expressing a phenotype, coding for a protein. Non-coding, what does that mean?
It's kind of part of the chromosome.
Uh-huh.
In that.
Oh, so you're a part of the chromosome, but by non-coding you mean that it has no function? Or I just want to make sure.
It might have function that we have not yet understood.
I like that. So, a unknown region of DNA. Okay, something along those lines. Anything else? Big word that starts with H. Really good. Heritable characteristics, heredity, right? That's the other thing that we normally associate genes and genetics with, right? The transmission of information from one generation to the next. Because it is true that DNA codes for either a protein or some sort of function, or even functions that we may know of yet, But the other very important way in which genes function in life is by transmitting that information to the next generation, because progenitors won't necessarily pass on the proteins or the phenotypes or the functions. What they do is they pass on the information for the new individuals to do that by themselves, right? So I want to home in on this idea here that genes code for a protein. And the reason I want to home in on that is because it's a very, very common slight misconception. And the idea here is that even though it is true that many genes code for proteins, there are many others that don't, right? So that is related to, by the way, I wanted to put into cartoons what we normally think genes might be at the molecular level, and that is that we go, what I just said, but this common yet slight misconception that DNA gets transcribed into RNA that is going to be translated into a protein. We see all kinds of cartoons, even in textbooks, the same textbooks that you guys are studying from will have drawings like this that somewhat imply that the genes are those regions of DNA that get transcribed into RNAs that get translated into a protein. And this is a depiction of the central dogma that some of you answered an assignment over the weekend addressing why this depiction is incomplete. But not only is that depiction incomplete in many ways, but it's also, again, slightly misleading in that it conveys this idea that DNA gets transcribed into RNA, all of which is translated into protein. And that is not entirely true, because there exists this very large family of what we call non-coding RNAs. So somewhat addressing what, even though you talked about non-coding DNAs, which is slightly different, and we can address that at a later point. But the point is that mRNAs, messenger RNAs, are the ones that get translated into proteins. But those are actually the minority of RNAs in any cell. The majority of RNAs that you'll find in any cell at any given time are non-coding RNAs, which I must say, it's a little bit of a misnomer, because non-coding RNA implies this idea that they have no function, that they carry no information whatsoever, which is not true. Non-coding is actually short for non-protein coding. That is true. These RNAs do not code for a sequence of amino acids that you'll find in a protein. They just carry information in the way of a series of nucleotides stitched together in a given sequence. They carry information that is expressed, manifested in other ways. So there is messenger RNAs that get translated into proteins, but there is many other non-coding RNAs, some of which assist in the translation of those mRNAs, and some of them you may have heard of before, like transfer RNAs and ribosomal RNAs. These are directly involved in the process of translation, even though they don't get translated themselves. They operate in translation, though they are not translated into proteins. But there are other kinds of non-coding RNAs. There are RNAs that regulate the abundance of other messenger RNAs within cells. We'll study some of these in more detail in a few weeks, but some of these non-coding RNAs can either stabilize certain messenger RNAs within cells, make them live a little longer, or actually actively destroy them and basically make them live a lot shorter. They destroy them, right? So those non-coding RNAs will regulate the abundance of messenger RNAs. There are some non-coding RNAs that regulate which and how much messenger RNA gets translated. So they will regulate down here during the process of translation. So there are non-coding RNAs that will either speed up or slow down the process of translation of certain mRNAs. And there is also, and these are just equally important, RNAs that regulate transcription. And I have a little exclamation mark there because these are newer. They've been around for as long as life has, but we know about them, or we've known about them more recently. Okay? These are RNAs that will facilitate or slow down the process of transcription itself. So they will go to where DNA interacts with RNA polymerases to transcribe certain genes, and it will tell these transcriptional machinery whether it should transcribe certain genes or not. Yeah.
Is it possible for, like, because mRNA goes back to DNA, is it possible for that to happen if there's like an error within the coding of the mRNA, or does that happen while the DNA is being made into mRNA?
Yeah, so the reverse transcription of RNA is not in response to errors in the transcription of that RNA.
So it's more than onions?
It's not even. It's a built-in function that retroviruses and transposable elements have. In their biology, part of their life cycle is to go from RNA molecule to DNA molecule and insert themselves in the genome. And it has to do with how evolutionarily for these types of molecules it was adaptive to take that route as opposed to live with information encoded in DNA. Right? For them, evolutionarily, it makes sense to go this weird route of being an RNA molecule that gets reverse transcribed, inserted in the genome, and then start being transcribed into several copies of RNA. Yeah.
Do we have to know what kind of RNAs functions as well?
Yes, and it won't be that hard because for each of these, there is really one type or two, and we'll address them in more detail when we get there. Yeah. And they have names that are somewhat self-explanatory after you've engaged with what they are, so it won't be too difficult, I promise. Any other questions, anyone? So, some of you may have touched on this in this assignment of completing the central dogma, but one of the misleading aspects of the most classical depictions of the central dogma is this idea that the only DNA that gets transcribed into RNA molecules that get translated. That is all a very linear path, when in reality, most RNA that gets transcribed from DNA is never translated. By some accounts, roughly 80% of the RNA present in any cell is non-coding. Once again, as in non-protein coding, not that they don't have any function. Actually, they're just as essential as messenger RNAs are. Cool. So here's a table that I leave here for your reference of the different types of RNAs and their function, right? And you will see some listed here, siRNAs, miRNAs, small nuclear RNAs. All of these we're going to discuss in a bit more detail. And you'll see when we get there that it's a pretty one-to-one relationship, so it's just a matter of memorizing 3 or 4 names and you'll be good. All right, so my second question to get some of those fluids going. Now that we're going to talk in a bit more detail about RNA, maybe a good time to point out some of the key differences with DNA. Briefly, we touched on the structure of DNA, and this is something that you covered in previous coursework, right? But up here we have the double helix structure of DNA, and down here we have sort of a molecular view of 2 base pairs between A and T and C and G in 2 opposite strands in anti-parallel orientation, and the base pair in between them, blah, 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 the biochemistry of DNA. So my question to you, what are some of the key differences from a structure, from a molecular point of view, between DNA and RNA? Give you a minute. Go. Okay. So what are we thinking? Just shout them out. Differences between DNA and RNA.
RNA has uracil instead of thymine.
So that famous exchange between uracil versus thymine.
RNA uses
Ribose sugar as opposed to deoxyribose sugar, right? So the D in DNA is for that deoxyribose. So the sugar in RNA has that hydroxyl group, whereas DNA lost it. So D-ribose, actually, that should go here. So D-ribose versus ribose. What else? Single-stranded versus double-stranded. Nice. Single-stranded versus double-stranded. Excellent. Anything else? Yeah.
DNA tends to stay in the nucleus, whereas RNA.
Oh, I like that. I did not have that in mind as I was putting my list together. But yeah, so in eukaryotes, DNA tends to be nuclear versus RNA is all over the cell, right? There is RNA in the nucleus when it's being transcribed, but then it goes into the cytoplasm to do all kinds of things. So I'm going to say all over. Just a slight detour. What else in eukaryotic cells you'll find some DNA? Mitochondria, and in plant cells, algae, chloroplast, right? These organelles that derive from ancestral prokaryotic organisms that were engulfed and kept some of their DNA. Excellent. Cool. Anything else? Yeah.
Maybe this is related to the sugars, but it is, never mind. You already covered it. The OH versus the H.
The OH versus the H. And I might as well address this issue now. In some textbooks you'll find, and it's not wrong, it's not false, in some textbooks you'll find that RNA can have catalytic function. And you'll read something about ribozymes, these enzymes that are made of ribonucleic acid. And it is very true that, they're a very special kind of RNA molecules, but it is true that they can catalyze chemical reactions. It's a bit of a stretch because the chemical reactions that they tend to catalyze have to deal or have to do with themselves in many ways, right? So some of these ribozymes have to do with processing other RNA molecules or themselves. And another sort of type of ancestral molecule that had that ability of cutting and pasting RNA molecules themselves are the ones that later, evolutionarily speaking, gave rise to things like splicing of eukaryotic prime-RNAs. So we're going to talk in a bit more detail about splicing on Thursday, but this idea that RNA has the ability to engage in chemical reactions with itself of sorts, and all that ties back to the presence of those hydroxyl groups at the 2' carbon position that makes these molecules chemically a lot less stable. And typically, less stable we associate with something kind of negative. If I told you that something is less stable, you're like, boo, RNA. But the reality is that lack of stability is what probably made RNA not be the preferred molecule for storing genetic information long term, but at the same time made it the preferred molecule to engage in all these interactions with various components of the cell. So we'll see that RNA not only has, in some unique cases, this ability to carry out molecular reactions, but also has the ability to interact with a whole bunch of things inside of cells. And part of it is due to its reactivity. All right, cool. So, yes, key differences with DNA, the base uracil is used instead of thymine. And another one is that ribose has an OH group attached to the 2' carbon, hence it is not a deoxyribose because it has the oxygen there. Now I want to touch on something that you mentioned, this idea that the RNA is single-stranded versus DNA that is double-stranded. RNA can or cannot base pair with other RNAs. What do you guys think? I see some heads nodding, right? Can, cannot. Can, yeah. Now, am I saying that RNA exists as a double-stranded molecule like DNA does? No, I'm not. Because what happens is, so does it or does it not have a secondary structure? That's what we'll discuss in a second. What happens is that this interaction of RNA molecules with other RNA molecules or themselves happens not throughout the entirety of the length of an RNA strand, but certain key stretches, right? So what happens is RNA can base pair with other RNAs, and yes, it does have a secondary structure. It's just that that secondary structure is not a long double helix. That's it. It's just collections of stretches of regions of RNA molecules that are either base pairing with themselves or others. And you can see that in these depictions of different real RNA molecules. So these three on top are ribosomal RNAs, and anytime that you see those stretches of what appear to be strings of dots facing one another, those are stretches of intramolecular base pairing of RNA molecules. So that means these molecules are base pairing with other regions of themselves. Hence the intramolecular, belongs in the same molecule. So those on top are ribosomal RNAs, and this is the structure down here of a typical transfer RNA. Again, a transfer RNA is almost nothing but stretches of secondary structure. And these secondary structures give these RNAs very specific 3D shapes, very specific configurations in space that will allow them to carry out their function. We'll see when we talk about translation that this L-like shape of transfer RNAs is sort of involved in the process of linking a series of key nucleotides that exist on one end of that molecule with whatever amino acid is loaded on the other end of that molecule. Okay? So RNA certainly can base pair with itself or with other RNAs, or even with DNA. And RNAs do form secondary structures more often than not, including, I don't have a picture here, but including messenger RNA. So if I asked any of you, next semester I'll do it, if I asked any of you to draw an mRNA molecule, and I wouldn't blame anybody for doing this, most likely you'll go something like this. And if I ask you to draw the process of translation, immediately you'll draw some ribosomes and then the protein being produced. And your mRNA molecule is that line, that squiggly spaghetti there, right? The reality is that many mRNAs exist within cells with secondary structures. So an mRNA molecule may look more like this, with one of those stretches of intramolecular base pairing, and then another one here with a little arm there, and then another one here. And these stretches of secondary structure do serve a role in the life of these messenger RNAs, because these are the secondary structures that may hide certain aspects of this mRNA molecule. They may facilitate the binding of certain key proteins to their molecule, and so forth. Okay? We'll revisit this when we talk about splicing and alternative splicing and how these secondary structures in mRNAs play a role. Questions, anyone? We're good? Okay. So, now we have a slightly more detailed depiction of the central dogma, where DNA is being transcribed into various kinds of RNA, many of which are non-coding. But this central dogma cartoon is still incomplete. And one of the reasons that it is incomplete or slightly misleading is this idea that an mRNA molecule will code for one protein. That is misleading because there is a series of mechanisms, both in prokaryotes and in eukaryotes, such that the sequence that is encoded in DNA and that gets transcribed into an mRNA through a series of mechanisms ends up being translated into different kinds of proteins. So in the case of prokaryotes, bacteria, what happens is there's genetic information. I need to choose my words carefully. There's genetic information in DNA that gets transcribed into what we call polycistronic mRNAs. These are long mRNA molecules that have in them information to be translated into different proteins. Right? Some of you may have learned about operons in bacteria and may remember your lactose operon, your tryptophan operon. And as much as you can remember this, those operons get transcribed into long polycistronic mRNAs, and they get translated into the 4, 5 proteins that are needed for that particular function. So polycistronic mRNAs give rise to several proteins, even though they come from one DNA sequence. In eukaryotes, there is a different flavor, but the essence of the message is the same, and that is, from the same sequence in DNA, eukaryotic cells often generate different mature messenger RNA molecules through a process that we know as alternative splicing, such that from the same region of DNA and from the same molecule of RNA that is originally transcribed from that region of DNA, you end up obtaining different proteins. Okay? Again, we'll discuss this in process in a bit more detail later. Notice that I, on purpose, left RNA out of this because I wanted to emphasize this idea that when you create that sort of link between sequencing DNA and the proteins that you generate from that region in DNA, there's something in the middle that makes it such that there's not one protein produced from that region of DNA, there is several different ones. Okay? Questions? All right. So, let's update our definition of a gene. Now we can go a little further from a region of DNA that codes for a protein. We can make it a little more generic and say it's a region of DNA that is transcribed into an RNA molecule. We're not implying messenger RNA anymore. It's an RNA molecule that carries out one or more phenotypes in a cell or organism. Now I'm using terminology we heard before, phenotype function, right? Including, but not limited, to being translated into one or more proteins. Right? So our definition of a gene is getting ever more complex. That's life. So now let's talk about transcription. I love this image. Before I used to have a photocopier, and that wasn't really the best metaphor for transcription. I like this better because here you can clearly see that there is an original template of whatever this document is. And then there is the process of transcribing the information in that original template to a new medium, in this case a piece of paper, most likely using the exact same language, except for those used for T's, right? That exchange of one letter for the other. So let's talk about transcription. So I'm going to ask everybody to sort of form small groups and add labels to these things. Here what we see is a cartoon of a region of DNA that is being transcribed and all these empty boxes, and I would appreciate that you slowly start adding labels to these things, and then we'll share the answers. All right, I hate to do this because I can hear there is still engagement with the exercise, but let's bring it back together and let's do this together. So let's go somewhat in order. What's this yellow blob thing? RNA polymerase. RNA pol for short. You'll see in textbooks, on my slides, and all kinds of sources. Polymerase abbreviated as POL, RNA polymerase. Good. What is this green molecule here labeled RNA? What kind of what name does it have? It's RNA, sure, but mRNA, right?
Is it ncRNA?
ncRNA, it could be either, right? It could be a messenger RNA or a non-coding RNA. We just don't know. So is there a more generic name for the RNA molecule being transcribed from DNA? Being transcribed from DNA? That's one kind of non-coding. This may be a term that you're encountering for the first time. Transcript. That's it. So in jargon, because we don't know if this is going to be an mRNA or any of the many kinds of non-coding RNA, we can simply call this thing a transcript. And that matters for the following reason. Most RNAs, be it messenger RNAs or non-coding RNAs, will typically undergo some sort of further processing. And this is actually a bit less so in bacteria. They're simpler organisms, certainly more so in eukaryotes. But all kinds of RNAs will go some sort of further processing. So when we're talking about the RNA molecule that is off the press, right, hot off the press from DNA, we generally call it a transcript. The cell hasn't really done anything to it yet that will make it a functional, mature RNA molecule of any kind. All right, so what's that end of the transcript? Is it the 5' end or the 3' end? 5'? That's right. RNAs, just like DNAs, get synthesized, get made from the 5' end to the 3' end with that polarity. So RNA polymerases will add nucleotides to the free 3' end of that growing transcript, such that the molecule grows in a 5' to 3' direction. Cool. We have DNA here unwound and the strands separated, and we give these strands names. So what's the name of this lower strand here, the one that RNA polymerase actually reads? The template. I gave it away in the title for the slide, but that's correct. That's the template strand. So template. And that is the one that RNA polymerase will use to stitch together nucleotides. So the way that this works, and we'll see that in the next couple slides, is nucleotides come into that template strand and bind to the template strand based on base complementarity. And once RNA polymerase senses that there is a good match between the incoming nucleotide and the sequence in the template, it will catalyze that reaction of stitching this new nucleotide to the growing transcript. Okay? And what do we call the other one? Coding. What was that? Coding. And here is a little bit of a it gets a little confusing here, because we call this strand coding, and it is the strand that will have the same sequence of nucleotides that you will see in the transcript, base for base, except U's instead of T's, right? And yet, molecularly speaking, the coding strand doesn't participate of this process at all. So we call it the coding strand as if it were involved somehow in dictating what that transcript should be, is pushed aside, and it has nothing to do with any of this. We call it coding simply because it happens to have the same sequence of nucleotides that you will see in the transcript. Why is that? Because the coding strand is base complementary to the template strand, and the transcript is base complementary to the template strand. So they happen to be complementary to the same strand, so they're going to be the same in sequence between them. Okay? In sequence and in polarity. So what is the polarity of the template strand? To the left, what would you guys write? This little box here. 3, that's right. So, oops. 3' to 5'. And that, once again, comes from the fact that base complementarity can happen only if the nucleotides are oriented in anti-parallel orientation. So if the RNA transcript is growing in the 5' to 3' direction, it has to be by reading over a sequence that is in the opposite direction, 3' to 5' in the template strand. And what is the orientation of the coding strand then? 5 to 3, the same as the transcript itself. All of these, I bet that all of you are understanding this. I ask you within a half hour, an hour, and you're confused again. Very normal. So, don't get anxious if you don't remember this by tomorrow. Just keep revisiting this idea, keep practicing. What is the orientation of the template strand, what is the orientation of the coding strand, what is the relationship between the coding strand and the transcript, and so forth. Yeah?
So the RNA is kind of like going from, let's say the yellow part is the
Polymerase.
it's kind of let's say a film tape projector, and the green, the RNA is like a film tape.
You
so
could think of it that way, yeah.
So it goes from the yellow bulb and then
As the yellow RNA polymerase is moving left to right, it's adding new green nucleotides to this growing RNA transcript. So the RNA molecule grows from left to right as the RNA polymerase moves from left to right.
so basically it moves through, right, and then leave a trail of.
That's right, yeah. And in fact, the next slide shows that in a bit more molecular detail. So now we have this big beige blob be the RNA polymerase. And what the RNA polymerase complex does is it unwinds both strands. It interacts chemically with the template strand. And it, position by position, it notices if the new nucleotide that came and base pair with the template strand is the quote-unquote right one or not. If it is, then it will link molecularly the end of this growing transcript with the new nucleotide. So it adds one nucleotide at a time. My recommendation to you guys is look online for videos showing these things. There's hundreds of them. Most of them are pretty good. Anything from Khan Academy to various YouTube instructional videos. We can talk about these things. We can look at static images, but most of those videos will do, even for verbal learners and audio learners, they will do a much better job than discussing these things based on pictures. Okay, so now we have on the screen, and we're addressing, this is a sort of an introduction to the processes of transcription and translation that we will discuss in more detail on Thursday and next Tuesday. But I wanted to quickly show you these two depictions. So normally we see all of these cartoons in which we have DNA on top being transcribed by the RNA polymerase that gives rise to an mRNA that gets translated into a protein. And then every once in a while, we find information that says, nah, not so quick. RNA polymerase will give rise to a primary RNA transcript, right? This molecule that is off the press from DNA, transcribed directly from DNA, but that needs some further processing. So it will be chemically modified on one end and the other. There will be some chunks inside of that primary transcript taken out in the process of splicing, right? And then you get this mature mRNA that can be translated. So my question to you is, which of these depictions is more accurate? I see a 2. Yeah? Yeah, that's right. We might be tempted in this spirit of, it's a little more complex, We might be tempted to think that this is an oversimplification, but it's not, because this is what happens in prokaryotes, right? This process of much more complex pre-mRNA processing is something that happens in eukaryotes. So this slide here shows how in bacteria we have various stages to protein expression. First, the cell needs to recognize a promoter, transcribe that region of DNA. There is chain elongation, meaning that the RNA transcript will be elongated to its full length. Then there is, in prokaryotes, a process called chain termination, and that's it. They're ready to translate. So it's not far from what this cartoon shows. DNA transcribes into a messenger RNA molecule that gets translated. So much so that most bacteria will do these two things simultaneously. As long as enough of an mRNA molecule showed up in the cytoplasm of a bacterium, the bacterium would start translating it in a process that we call co-transcriptional translation. It's translation that is happening at the same time as transcription. Okay? What is shown on the right side of this image is also true. In eukaryotes, you have promoter recognition, transcription initiation, and chain elongation. There is an extra step of RNA processing that involves all of these capping and polyadenylation and splicing, all of which we're going to discuss on Thursday, so that we collectively call RNA processing, and then you get your mature mRNA that can be translated. Okay? So both true. It's just that one in prokaryotes, the other one in eukaryotes. So now, before we go on to Thursday and to discuss the process of transcription in a lot more detail, let's talk about gene structure, because there is always a little confusion about, we started out with a conversation of what a gene is, and we evolved from saying a region of DNA that codes for a protein to a region of DNA that gets transcribed into an RNA molecule, right? That can serve a number of functions, some of which may be being translated into a protein, but there may be other functions that have nothing to do with being translated into a protein, but facilitating other processes inside of cells. So that kind of begs extending this exploration of what a gene is into gene structure. So what we know about genes is that they have various components. They have various parts to them. Obviously, there is that region that gets transcribed into an RNA molecule, right? We call that the coding region of the gene, and it's the portion that is transcribed into RNA. We talked about this already. The template strand is the one that RNA polymerases read and walk on. The coding strand stays out of the way, but has the same sequence of the RNA transcript. They're guilty by association, if you will, right? So they have the same sequence as a transcript, even though they have nothing to do with the actual biochemical process that generates that transcript. So the coding region is what actually gets transcribed into an RNA. One key component of all genes is, and I have it here on a slide dedicated to it, is the actual start of transcription site, or TSS, transcription start site. That's how it was originally named. It is also known as the +1, because that is the very first nucleotide that you will see in the RNA transcript, right? So it corresponds to the first DNA nucleotide that is transcribed into RNA, and it is often denoted as a bent arrow or angle arrow pointing in the direction of transcription. So when you see schematics of genes that are simplified for much easier understanding, you'll see a straight line denoting DNA and one of these bent arrows indicating that that's where transcription of that region begins and in which direction it goes. I want you to notice this because it's an important point. It may be tempting to think that the template strand for one gene is the template strand for all genes in a given DNA molecule. And that's not true. What the template strand is and what the coding strand is dictated by where transcription begins and in which direction it goes. So for this gene here on the left, the promoter is going to the right. What that means is that the template strand will most likely be the one here, the lower strand. That's where RNA polymerase will read in the 3' to 5' direction to produce a transcript in the 5' to 3' direction. But for this gene here, it happens that the TSS is pointing in the opposite direction. What that means is RNA polymerase will go backwards, will go in that direction, reading the upper strand as the template strand, and transcribing, again, a molecule in the 5' to 3' direction by reading a template strand in the 3' to 5'. So which DNA strand serves as a template depends on where that TSS is and in which direction RNA polymerase takes off. Okay? So how does RNA polymerase know in which direction it should go? Let's say the RNA polymerase lands on the TSS. How does it know it should go one way or the other? You had some thought?
The coding strand of it, so like AUG for the...
AUG is for translation, comes later.
I can't remember if this was Professor familiar with the Tata box.
The Tata box is related to this. We'll discuss the Tata box in detail on Thursday, but do you remember what a part of what the Tata box is normally? A promoter. So that's the other key element of all genes. We call it a promoter, and the promoter sequence is something that sits just upstream of the TSS. And it is essentially a series of signature sequences in DNA that have the ability to recruit RNA polymerase and transcription complexes to a given location in DNA and tell them in what direction to go. So not only do they bring RNA polymerase to the TSS, but also tell them which way they should go. And from there on, RNA polymerase, being the quote-unquote stupid molecule it is, it will just walk on the template strand that is 3' to 5'. It just doesn't know in which direction it's going. It was just placed on DNA and oriented in such way that it will follow the 3', 5' direction. So how the RNA polymerase is placed on that location in DNA, looking one way or the other, is what the promoter does. The promoter, through these very specific collection of key sequences, we call them consensus sequences, will tell RNA polymerase where to bind in DNA and which way to look. Yeah, I think I said everything. So those are promoters. And in this example that we had here, why RNA polymerase will go in one direction or the other is all dictated by the promoter. So those key sequences in the left gene are such that they will place RNA polymerase at this TSS and have it look to the right. The promoter in this second gene will place RNA polymerase at this TSS and have it look to the left. And from their point onwards, RNA polymerase will simply follow the 3' to 5' template strand because that's the only thing that biochemically it can do. Yeah.
Is this part of the reason why the same section of DNA can code for multiple proteins? Because if it goes in one direction, it might...
It's rare. So what you're suggesting is that this region here, which I may have alluded somewhat with my schematic, what you're suggesting, this region here could be transcribed one way to give rise to one protein and another way to give rise to another protein?
Yeah.
Absolutely nothing theoretically against that. Evolutionary speaking, what happens is that creates such constraint on how much both sequences can evolve, because imagine that anything that you do to this strand here will be reflected on what happens to this strand here. So it's rare that you can tinker with one sequence without breaking the other,
That makes sense.
right? So evolutionary speaking, there's a lot of pressure for not having that. And genes may go in both directions, but they tend to finish before the other one begins, in a way. So I didn't draw it, but since you asked, I'll do it. So this gene here may go from here to here, and this other gene down here may go from here to here. So they're going in opposite directions, but it's rare that they will have overlap. Not impossible, but rare. Any other questions, anyone? So, quick question for all of you. Does the promoter get transcribed?
No.
No, it doesn't. It tells RNA polymerase where to begin, but it kind of sends it in the opposite direction. It says, start here, go off that way. The promoter is upstream of the TSS, meaning that it's just before the first nucleotide being transcribed and all the other ones. So the promoter is a key component of genes that does not get transcribed. So that right there is already throwing some shade at our simplistic definition of a gene, region of DNA that gets transcribed, because it turns out that some key components of genes do not get transcribed. The promoter is one of them. The other one that happens mostly in prokaryotes when it comes to messenger RNAs, to protein coding genes, but it applies to eukaryotes as well for some non-coding RNAs, is the termination region, essentially a sequence in DNA that gets partially transcribed into RNA, but it also serves as some sort of signal that we'll study in a bit more detail on Thursday that tells the cell, you know what, you should stop transcription here. So part of that termination region gets transcribed, but not all of it. Some of it never gets transcribed either. Okay. In eukaryotes, I emphasize that non-coding RNAs can have termination regions like prokaryotes do, but all protein coding genes, those that are transcribed into mRNAs, don't have active termination. Instead, what they have is something we call a polyadenylation signal. It's a signal that gets transcribed into that primary RNA transcript, and that as part of that processing of the RNA that eukaryotic pre-mRNAs need to go through before they can become mature and be translated, as part of that processing, the cell recognizes that polyadenylation signal and knows that it should cut that primary transcript there at that position, and as part of a process, like almost an enzymatic reaction, except that it's a complex one, it will add a string of adenines to that 3' tail of the RNA. So you end up with an RNA that is truncated and polyadenylated, meaning it has had a bunch of A's added to them. Very common mistake to think that the poly-A tail that you will see in any mature eukaryotic mRNA is coded in DNA, meaning that in DNA, eukaryotic protein coding genes at some point will have T in the template strand, which will result in a poly-A being transcribed. No, doesn't work that way. So there's whatever sequence that gets transcribed, and somewhere within that sequence there is a poly-A signal that serves as as the name indicates, a signal for some enzymatic machinery to come in and do those two things: cut the the RNA molecule and add a bunch of A's. Okay? Why cells do that and how they do that, we'll discuss on Thursday. The point that I wanted to bring home with this slide is that, like the promoter sequence, there's many aspects of the end of an RNA molecule that are not directly transcribed from DNA, right? So there are components of DNA sequence that never make it to the final mature RNA molecule that is transcribed from them. And yet, they're essential for telling the cell what to do with these various regions of DNA that need to be transcribed to RNA. So they're essential components of the gene. It's just that they're not necessarily reflected in the sequence of nucleotides in those transcripts.
Transcription factors part of the gene structure?
No, because transcription, so the question here is whether transcription factors are part of the gene structure or not. No, because transcription factors are proteins. When we talk about gene structure, we're talking different regions of the DNA molecule itself, the DNA sequence. Then there is the stuff like transcription factors that interact with these sequences, read these sequences, and do things when they see those sequences. But they're proteins. Transcription factors are proteins that interact with DNA in very specific manners that are based on sequence. But gene structure, we're talking about regions of DNA and how they relate to whether or not they get transcribed into RNA or not.
So the ending of the mRNA shows as A, and the ending of the template shows as T.
It does not. What I emphasize is that it is a common mistake to think that the template strand will have T in it. It doesn't. It doesn't. What happens is after the template strand was transcribed into the primary transcript, there is somewhere in there a short polyadenylation signal and that is recognized by one of these complexes of proteins that come together, see that, and they say, okay, we know what our job is. We need to cut this RNA molecule and start adding A's. But it's not based on template. It's not part of transcription. It's post-transcriptional.
The RNA just edits on itself.
Yeah. It's this machinery that works with the RNA, but yeah, it's not based on DNA sequence. It's a process that is inherent to this polyadenylation machinery. Cool, so I'd like to finish with one last time I promise, what might be like the last, most elaborate version of a definition of what a gene is. So it's a region of DNA that contains sequence that is transcribed into an RNA molecule that carries out one or more phenotypes in a cellular organism, including, but not limited to, being translated into one or more proteins, plus any non-transcribed sequence that is used by cells to know when, where, and how much said RNAs should be transcribed and how they should be processed afterwards. Right? So we're far from that simplistic definition of a gene as region of DNA that codes for a protein, because it's a region of DNA that codes for a given RNA transcript, plus any information that the cell needs to know when to transcribe that region of DNA, how much to transcribe that region of DNA, where, as in multicellular organisms, which cells should transcribe that region of DNA, right? And I said when, how much, and where. I think I hit them. And any sequence that, like what I was saying about the polyadenylation signal, any sequence that gets originally transcribed to the primary transcript, but is not part of the final mature mRNA molecule because it simply indicates further processing. And this is something that we'll touch again when we talk about splicing. There is sequences in that primary transcript that won't be there in the final product and yet carry information as to how the cell should deal with them. Okay? All right.